Protein synthesis is composed of three main steps: transcription, RNA processing, and translation. The purpose of protein synthesis is crucial for the genetic diversity of all of our cells. Because each cell within our body contains the exact same genome, in order to produce differentiated cells and produce our tissues and organs, protein synthesis is required to code for specific polypeptides that will regulate cell traits and genes. The first step of protein synthesis is transcription. Transcription is vital for the building of amino acids because DNA cannot be used to directly code for these amino acids. DNA must stay within the nucleus and cannot be taken to the sites of protein building. In order to get the information from DNA to where it must be synthesized, it is transcribed as RNA, specifically single-stranded mRNA. The first step within this process is initiation. It begins with a strand of DNA. Within that strand of DNA, there is a promoter, which is the start site for transcription, and a terminator, which is the end site for transcription. To begin transcription, the enzyme RNA polymerase will attach to the promoter. In eukaryotic cells, the promoter contains a TATA box. Before RNA polymerase can bind to the promoter, several transcription factors must bind to the TATA box. The next step is elongation. The RNA polymerase moves downstream, unwinding the DNA and elongating the RNA transcript by joining together RNA nucleotides complementary to the DNA template strand. This occurs in the 5' to 3' direction. The final step is termination. In termination, the RNA transcript is released when it reaches the termination point in DNA. This is often an AAT-AAA sequence. After transcription is complete, RNA processing must occur before releasing this mRNA strand into the cytoplasm. At the 5' end of the mRNA strand, an enzyme adds a cap made of modified guanine triphosphate. The 3' end is also given a poly-A tail, which is made up of about 200 adenine nucleotides and constructed by the enzyme poly-A polymerase. These are important in signaling that the mRNA strand is mature, preventing degradation, and helping the ribosomes attach to the 5' end of the strand. Another alteration must be made to prepare the mRNA for translation. The mRNA strands currently are made up of introns and exons. The exons code for polypeptides, however, the introns are non-coding. Splicing must occur to take out the introns. snRNPs join with other proteins to produce a spliceosome, which catalyzes the process, removing introns and joining exons. Once this is complete, RNA processing is finished. After RNA processing is complete, the strand has exited the nucleus and translation can occur. Translation is using the mRNA to synthesize a polypeptide. The first step in this process is initiation. Initiation begins when a small ribosomal subunit binds to the molecule of mRNA at the 5' end. An initiator tRNA with the anticodon UAC binds with the start codon AUG. This tRNA now carries the amino acid MET. The large subunit then arrives to complete the initiation complex. The ribosome contains three main sites: the E site, which is the exit site, the P site, which is the peptidyl tRNA binding site, and the A site, which is the amino acidyl tRNA site. The initiation tRNA is currently in the P site. The next step of translation is elongation. Codon recognition occurs here, in which the anticodon of incoming tRNA pairs with the mRNA codon at the A site. From here, a peptide bond is formed by an rRNA molecule of the ribosome, catalyzing the formation of a peptide bond between the amino acid group of the new amino acid in the A site and the carboxyl end of the growing polypeptide in the P site. Translocation then occurs in which the tRNA in the A site is moved to the P site. The empty tRNA was that was in the P site is moved to the E site and is released. Finally, termination occurs when the ribosome reaches a stop codon. A release factor binds to the stop codon and releases the polypeptide from the ribosome. Now we'll look at gene regulation. Regulation will be modeled using a lac operon within the E. coli bacteria cells. Operons are groups of genes that code for proteins with related functions. In these bacteria cells, transcription and translation occur at the same time. Gene regulation is the process of turning genes on or off depending on the amount of substance and whether it needs to be broken down or maintained. The lac operon is inducible and is made up of an operator, a repressor, a promoter, an inducer, regulatory genes, and specific operon genes. The regulatory gene, LAC-L, produces an active repressor that binds to the operator region. When the operator region is blocked, it prevents RNA polymerase from transcribing the structural genes that control the breakdown and uptake of lactose. When lactose is present, it binds to the receptor protein. It binds to the receptor LAC proteins, changing its shape, which causes it to release from the DNA. Because of this, when lactose is present, the lactose digesting enzymes can be transcribed. Lactose here is an inducer. The three structural genes of the lac operon code for different proteins. LAC-Z codes for beta galactosidase, which breaks down lactose into glucose and galactose. LAC-Y codes for the channel protein permease to transport lactose into the cells. And LAC-A codes for transacetylase, which detoxifies other molecules that enter through permease.